Evening everyone, this is um, a quick tutorial, as it were, quick lesson on DNA structure. So the structure of DNA, how the structure is related to its function, and then perhaps a little bit of analysis of some data for DNA. This all relates to booklet 7, so DNA and genetic diversity. It's the first few pages, 2 or 3 pages on nucleic acids. Okay, so firstly, where does the DNA exist? Well, this is a eukaryotic cell. Looks like a cheek cell to me, and inside that cheek cell is a nucleus. Now, if you look really carefully at this image, you will see that those chromosomes exist here as these X-shaped chromosomes. That's pretty typical of a diagram you might see in a textbook, but it's a little bit misleading. Okay, so let me draw a little diagram for you here. Okay, so when we see these X-shaped chromosomes, like so, what we're actually looking at is a chromosome that is comprised of a pair of sister chromatids. Okay, so there's one sister chromatid and a second sister chromatid. Now it only exists like this after DNA replication has taken place, and in fact, even more specifically, it's just before, so just prior to cell division. So that could either be prior to mitosis or meiosis. Okay, so the only reason you'd ever see it like this is in this, what we would refer to as this condensed form, is because the cell is just about to enter either mitosis or meiosis. Okay, now if I go back to here, even this diagram is also a little misleading, because again, this shows the condensed form of the chromosomes, but it's perhaps a little bit more realistic about what you would expect to find before DNA replication. It shows the homologous pairs. So chromosomes come in pairs. One paternal chromosome, so a chromosome that was inherited from the father, and one maternal chromosome inherited from the mother. And those pairs are what we call homologous pairs of chromosomes. Okay, so a homologous pair of chromosomes is, well, we'll look at the definition specifically in a minute, but it are two chromosomes with the same genes at the same loci, and that's shown in this diagram here. Okay, so we can see each of these bands, they're in the same position. The position is what we refer to as the loci, or many loci, or an individual locus. And the position of those genes is the same on the paternal chromosome as it is on the maternal chromosome. So they have the same genes at the same loci. And there are two of them. So the definition, as we'll come back to later on, is two chromosomes with the same genes at the same loci. Okay, so what is a gene, you're probably asking? Well, a gene is a section of DNA with a specific sequence of bases. Now I'll come back to what these bases are later on, but the base essentially, or the sequence of bases, is what we refer to as the genetic code. And that code, genetic code codes for a particular protein or a particular sequence of amino acids that form a polypeptide. Okay, so the sequence of amino acids that form the polypeptide, that's determined by the sequence of bases in the DNA on a chromosome. Okay, so genes are made up of DNA, deoxyribonucleic acid. So let's go to this next slide. Now it shows that chromosome existing again as this pair of sister chromatids held together by the centromere. And that's the same as on this diagram here. Okay, so the pair of sister chromatids held by the centromere. That centromere holds the two DNA molecules together. So what we've got to imagine in here is just one wound up molecule of DNA there, which is identical to a wound up molecule of DNA in here. Okay, they are the product of DNA replication. So one sister chromatid contains DNA which is identical to the second sister chromatid. So DNA itself is made up of these nucleotides. The nucleotide is the monomer, and it's comprised of three parts. So let's go have a look at the three parts of a nucleotide. Let's go for a rainbow colour. Okay, so the three parts of the nucleotide are the inorganic phosphate group. And that is bonded to a five-sided sugar. So I'm going to refer to it here as a pentose sugar. But if this is DNA, that pentose sugar will be deoxyribose. And then there is what's sometimes referred to as an organic base. I tend to call it a nitrogenous or nitrogenous base. It's nitrogenous because it contains nitrogen. Okay, now there are four different types of bases in DNA. They are adenine, guanine, cytosine, and thymine. Okay, and it's the sequence of these bases which determine the sequence of amino acids in a polypeptide or in a protein. Okay. Now, RNA is very similar to DNA in many ways, but there are some differences. It still has an inorganic phosphate group. It still has a pentose sugar. The difference is RNA contains the pentose sugar ribose. Hence the name RNA, that R from ribose and the D for deoxyribose. So when in a question, it may ask you to identify one of these three parts of the nucleotide. Now you need to see in the question, it was like blah, 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 something, DNA, blah, 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 identify or give the names of the three parts. If it says DNA in the stem of the question, you must refer to deoxyribose. If it says RNA in the stem of the question, ribose. There's one other difference with RNA. Okay, it still has 4 different bases, but in RNA, so these are the DNA nitrogenous bases. In RNA, it still has adenine, guanine, cytosine, but thymine is replaced by uracil. Okay. Now, we'll talk more about the different forms of RNA. There are 3 forms that you need to be familiar with, which are mRNA, tRNA, and rRNA. We'll talk about those 3 forms when we come on to look at protein synthesis. Oh my goodness, in future lessons. Okay, so this is the monomer, known as a nucleotide. It forms by condensation reaction, so condensation reaction between the phosphate group and the deoxyribose, and then a condensation between the deoxyribose and the thymine. Okay, and that forms 2 bonds. This bond here is an ester bond. This bond here is a glycosidic. It's probably not going to be too important, that. But now, mention that. So, DNA exists in this, you've heard this phrase before, a double helix. Double just meaning that there are 2 chains or 2 strands of polynucleotides. Helix meaning it's twisted. Helix, that's really useful because it makes it condensed, i.e. it's smaller or compact for storage. We'll look at some of the other features of the double helix later on. Okay. Between those 2 strands are the pairs of bases. Okay, so here is a single strand, and we can see that the single strand, it goes phosphate, deoxyribose, phosphate, deoxyribose, blah, 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 phosphate, deoxyribose, phosphate, deoxyribose. This part here is referred to as the sugar phosphate backbone. And the sugar phosphate backbone is joined by these covalent bonds. And actually the linkage between one deoxyribose or phosphate and the second deoxyribose is known as a phosphodiester bond. Phospho, because obviously it contains inorganic phosphate. Diester, well, we mentioned that there are esters, and there are obviously 2 of them, one there, one there. So phosphodiester bond, which links those phosphates and deoxyribose sugars to form the sugar phosphate backbone. Now it's double stranded, so that sugar phosphate backbone is paired up with another molecule, another sugar phosphate backbone, bases extending out from that, and we can see here what we're beginning to get, which is a different colour, a bit thinner, I think. Between here are the complementary base pairs. And this is one of those phrases in biology and A-level biology that comes up all of the time. Okay, complementary base pairs. We've talked about things being complementary to each other before. So if they have a complementary shape, it means they're not the same shape, but they fit together. The complementary base pairs in DNA form between adenine and thymine, and 2 hydrogen bonds form between those, and cytosine and guanine, and 3 hydrogen bonds form between those. Then we've got another adenine, thymine, 2 hydrogen bonds, another cytosine, guanine, 3 hydrogen bonds. That produces that sugar phosphate backbone. Remember, the sugar phosphate backbone joined by phosphodiester bonds. And there are 2 of them. The 2 sugar phosphate backbones protect the sequence of complementary base pairs between them. Okay, and that makes the molecule more stable. There we can see the complementary base pairing. So adenine and thymine, we can see 2 hydrogen bonds between those bases, 3 hydrogen bonds between the cytosine and the guanine. Now, one other thing you may notice about this diagram is the relative directions of the 2 polynucleotide chains or polynucleotide molecules. So we can see that they run in opposite directions. The phrase we use here is they are anti-parallel. Okay, anti-parallel means that they are they run in opposite directions. Okay, this is this diagram again shows the phosphodiester bonds that form between the phosphate groups and the deoxyribose, and then it shows those hydrogen bonds that form between the nitrogenous bases. Okay, so the genetic code is the sequence of nucleotide bases. And that sequence of nucleotide bases determines the sequence of amino acids in a polypeptide. And that's really important. Okay, so the nitrogenous bases, their sequence determines the sequence of amino acids in a polypeptide. One really common error, and I hope none of you ever make this error, is to say that the DNA is made up of a protein. That is just absolutely wrong. The DNA, or the sequence of nucleotide bases, may determine the sequence of amino acids in a protein, but they are not the same thing. Okay, we'll just skip through to here. Here we can see again those complementary base pairs. You can see the 2 sugar phosphate backbones, and we can see the relative direction and that anti-parallel nature of the 2 chains, which run in opposite directions. Okay. Just do this a second. Okay, one thing that you may be asked to do is determine, let's see if I can find it. Ooh. Ignore this, sorry. It's to either determine the number of bases or the percentage of bases present in a molecule of DNA. Okay, we know that adenine and thymine are complementary to each other. So if adenine and thymine are complementary to each other, there must be the same number. Okay, guanine and cytosine are also complementary to each other, so they must have the same number. Okay, we've got two different strands, they're all combined, so we know that guanine and cytosine are complementary. Then we know that there are 17 base pairs altogether. 17 minus 5 plus 2 plus 6 must mean there are 4, and therefore there must be 4 thymines there as well. Okay. So the importance of DNA. Now, if we just have a look at the notes, we've been through that. Oh, we've been through that. Okay, so there are some important functions. Stop there. Some important functions of DNA. One of them we've mentioned already. So, genes code for polypeptides. More specifically, what we mean there is the, as we said before, the nucleotide base sequence codes for a polypeptide. They determine inherited characteristics. By characteristics, perhaps a better term to use there is a phenotype. This makes it a bit thinner. Is a phenotype. So a phenotype is the visible expression of a gene. So it's the genotype, i.e. the genes that are being expressed, plus the influence of the environment. And that's what provides these characteristics. Okay. This provides genetic diversity. So different versions of genes are known as alleles. Okay, so those alleles are produced by mutations to a gene. So the sequence, the nucleotide-based sequence, may change to produce a new allele. And different alleles create different gene phenotypes, and that provides that genetic diversity or variation that we see in populations. Another really important feature of DNA is its ability to self-replicate. Goodness, that's not very good, is it? So, its ability to self-replicate. Now, the reason for that, let me find a diagram over here, is all to do with the base pairing. Let's find this one here, in fact. Okay, so a single strand of DNA in the presence of some free DNA nucleotides. So we've got some adenines and some thymines and some cytosines and some guanines. Those free DNA nucleotides will spontaneously pair up with their complementary base pair. So thymine would spontaneously, without any influence of any enzymes, without any help, pair up with adenine. Guanine would spontaneously pair up with cytosine, and therefore cytosine would spontaneously pair up with guanine, and then another guanine with cytosine down there. This idea that they are spontaneous gives DNA one of its really key features, and that's its ability to self-replicate. Because it's spontaneous, that pairing up of the complementary bases, we can refer to it as being self-replicating. Now that's really important because it's important that the DNA produces a copy of itself. It replicates prior to cell division, so prior to meiosis and mitosis. That variation, the last little thing, in combination with its ability to self-replicate, well, the variation, there's different alleles. Some alleles will be beneficial, some will be detrimental. The ones with the beneficial alleles for a particular selection pressure will be selected for, and they will survive, reproduce more often, and pass on more of those alleles. That's this idea, the concept of natural selection. So one of those fundamental, if not the fundamental concept in biology. So natural selection leads to speciation and evolution over many generations, over a longer time period. Okay, other features then, other structural features and how that relates to their function. Well, we've mentioned this already. Sugar phosphate backbone gives the molecule lots of strength. The coiling of the DNA, i.e. the helix, makes it compact, better for storage, and actually able to fit in the nucleus. If you combine that with some of the DNA wrapping around those histones that we mentioned earlier on, that allows a molecule of DNA that may be 30 centimetres long to fit inside a eukaryotic nucleus. Okay, it's a double helix, so 3 features here. Each strand acts as a template for replication. The double helix, where it protects the genetic code, the bases on the inside are protected by the sugar phosphate backbone. And it also makes the molecule more stable. It's really large, so lots of information can be stored in a molecule of DNA. Lots of hydrogen bonds. Collectively, those hydrogen bonds give the double helix lots of stability. However, and that's really good because it prevents that code from being disrupted. So it prevents damage to the code. And it also means that when the DNA replicates, because of those hydrogen bonds and the complementarity between the bases, the code is preserved. So that's collectively. But individually, those hydrogen bonds are actually very weak. So it's quite straightforward with an enzyme to break those individual hydrogen bonds for replication. So when DNA copies itself, so the copying of the DNA, and for transcription, which is the first process in protein synthesis. The sequence of bases, well, we've mentioned this already, codes for the synthesis of specific proteins or specific polypeptides, i.e. the amino acid sequence in a polypeptide. And because of the complementary base pairing, well, it is self-replicating, and it enables the information to be replicated really accurately. And that's because adenine will only pair with thymine, cytosine will only pair with guanine. Right, and that's everything really for the structure of DNA. So we need to know the structure as a monomer, so a single nucleotide. So a phosphate group bound to the deoxyribose, bound to a nitrogenous base. And we also need to know the double helix structure. Let's see if I can draw this with my new pen. The easiest thing in the world. And there's base pairs between them. So we need to know those the structures as the monomer and the polynucleotide, and then the double helix as well. So we've got polynucleotide, just a single one between those. We've also looked at how the DNA structure is related to function. And we've looked at some of that data about DNA. Let's just do one more example of those. Okay, for example, knowledge about one strand. Let's see the answer. So it's able to you're able to analyse the composition of DNA by knowing the information about one strand, can work out the information about the other complementary strand. So if we know that DNA has thymine, and that thymine is 22%, well, there must also be 22% adenine. If we know that, 22% plus 22% makes 44%. The remaining bases must be 56%. Cytosine and guanine have got to be equal because they are complementary base pairs. So 56 divided by 2 is 28%. So being given the information about just one of the bases in a double helix of DNA enables you to calculate the rest. Okay, and that's everything for this video. If you've got any questions, leave them in the comments or you can email me. I hope that cleared stuff up from this morning. Okay, thanks very much.